Good morning, everyone. My name is Jason Kolbert. I'm the head of research at Boral Capital. I'm very excited this morning to have Dr. Chris Missling, someone who I've known before he was the CEO and founder of Anavex. Chris is, I've known Chris, I've known you a long time. I think you're very, very smart. I also think you have a lot of integrity, and I believe you're a very honest person, very sincere person. And that's one of the reasons why I'm so bullish on Anavex. I believe the stock has tremendous potential. And more importantly, I believe the therapeutic has tremendous ability to, I don't want to use the word rescue, but maybe stabilize patients who are facing dementia and CNS maladies, not just Alzheimer's disease, but there is a host of other indications. The purpose of getting together today is to talk a little bit about what's happened in Europe and why did European regulators decide not to approve the Alzheimer's drug, and what's going to happen going forward. And then let's talk a little bit about where you are with the US. You had what I consider to be some very good news the other day, talking about interactions with US regulators. And I want people to decouple a little bit some of the events in Europe with what the pathway looks like in the US, because there are different pathways. The US doesn't follow Europe. Europe doesn't follow the US, although in the end, I think they both end up in the same place. And my bet as a biotech analyst is that that place is approval. This is an unmet medical need. It is a desperate need. The approved drugs today don't do much to change the disease trajectory. And I think one of the promises of AVXL 273 is it could potentially change that trajectory. And that's a disease modifying agent. Another area too, and I firmly believe this, is that the drug has shown itself in thousands of patient exposures to be very, very safe. And again, this harkens back to some confusion out of the European release, which suggested that it wasn't safe. So why don't we start right there and just give me some background about what happened in Europe? Thanks, Chris. Thanks for joining me this morning.
Welcome. I appreciated the opportunity. So maybe I'll start with where we are with Europe. So we are in the discussion of what also preceded the two recent antibodies in Alzheimer. They also had been in the same situation where in the first round of review, they did not get the recommendation from the EMA, and we basically share the same situation. But they were then able to get the approval in the re-examination. And we are proceeding in the same fashion. So we're going now in the re-examination with the EMA, which will start soon. And you will get another pair of rapporteurs, which is two members from the European member states, which will be assigned to review our case again. And it's basically a couple of fresh airs to look at this. And
So,
what
Chris, very important point. What you're saying is that you're kind of on a pathway following a historical precedent where the EU is not so quick to approve the first time around, that there's often a second round. And what I hear you saying is in the second round, when you have a new reviewer, it's like a fresh set of eyes looking at the data, and therefore you have potentially an improved chance being approved because by that time, the second reviewer is now addressing not only what the issue was with the first reviewer, but the company's response to it. Is that fair?
that is fair because also we believe that the procedure is very complex. And there's so many documents and data points which make this process maybe not efficient enough to allow for the CHMP, for the review by the EMA, to comprehend the totality of the data to grant the approval right away. So almost you need a second session to allow the data to be understood, to be presented, and it's maybe an issue of the process. And that is, in a way, a example of the reason why this takes two stages, maybe. So that's a possible other
Good.
reason
Fair
why
enough. Fair enough.
that is the case.
So, can we talk a little bit about safety? Is this drug safe? What was the issue that the European regulators had? And one of the things that I was fascinated by was you used an analogy to me when the data first broke. We were talking about nitrates and food, hot dogs, I believe. So, put that all together for me.
Yeah. So when you have a drug manufacturing process and you have a nitro group in your drug, which many drugs have, and we have that as well, there is a concern that there's impurities in the procedural of the drug manufacturing, which might lead to a level which is similar to what is in the salami or in the prosciutto, which is cured meat, red meat, and that is the nitrosamines the nitrosamines. And if they have a certain elevated level, they have been associated with potentially causing, cancerogenic features. And for that reason, there's a threshold worldwide for manufacturing of testing those impurities. We have in our manufacturing process, which is run by the largest US manufacturer, by the way, we are very clean on that threshold. However, the EMA has reduced these threshold levels even further, which are very conservative to begin with. And we had, during the submission, retest with these more sensitive diagnostic measures the level of impurities in our drug process, manufacturing process. And we just recently completed that testing and showed that the presence of impurities are below the levels required by EMA standards. So that basically it is a moot point for us going forward.
Great. And just one other point you made to me was that if I was to have a salami sandwich or a beautiful piece of prosciutto bread, my nitrate levels would probably exceed what the EU standard is,
That's
at least
exactly
in
right.
my blood an hour after I ate it.
That's exactly right. That's
Okay,
correct.
good. So safety is not an issue.
We
So then
believe it's not.
good.
Yeah.
And that's very helpful. So talk a little bit to me about efficacy and about what's it take to prove efficacy. And obviously, Alzheimer's is a tricky drug. There is talk about what is the right endpoint and where do you fit in terms of that endpoint. And couple that, I know you will, with the data that was submitted with the application versus the totality of data that's going to come into play in the second review.
Yeah. So we had in our case pre-specified a co-primary endpoint ADAS-Cog 13 and ADCS-ADL. It turned out that the second co-primary endpoint, ADCS-ADL, was invalidated by science. Over the duration of the trial, publications came out, guidances were also came out from EMA and also from FDA that the ADL, the quality of life, activities of daily living, and that's what it stands for, is not significantly changing for early Alzheimer population. For early Alzheimer population, that scale is not sensitive enough to use as an endpoint. And we had that pre-specified because that's when we started the trial, that was considered the standard endpoint for function. However, we included also what is today the standard for cognition and function in one endpoint, which is the CDR sum of the boxes. And we had that
I'm
as
sorry, say that again. It was the
CDR sum of the
CDR.
boxes, CDR-SB. And that was the key secondary endpoint. So what we are now arguing is that if you are uh, scientifically able to confirm that a scale is not anymore useful for a certain population in a study, you can make the case that if you are correcting that with the adjustment by making the bar higher for achieving significance, because the procedural requirement was to have the significance measured at 0.05 as a p-value and lower. So if you now say because of the ADCS-ADL was invalidated, you just basically remove that and make the bar higher by the significance level has to be 0.025, or the significance level have to even higher by using three endpoints dividing by 0.05 by 3 is 0.0167. You make the case, what is called an adjustment for conservative correction, and you have to then show that those endpoints selected, ADAS-Cog 13 and CDR of the boxes, are below this higher threshold. And that is the case in our trial. So the argument is the trial has met the pre-specified endpoints by this correction, by this adjustment, because it was not known to the company that the ADL would not change in this early population. And by the way, the ADL was trending in the right direction and also was significant for the sub group or the population in the trial, which was the genetically defined population, AB clear 2 and 3, which allows also to make the case if we would go into that population in our resubmission or re-examination, we would have the ADCS-ADL significant. So we would have the co-primary endpoints met, as well as the CDR from the boxes, as well as the other endpoints. So this would be then allowing to have the discussion going in that direction, which also showing with very high significant improvements of the scales compared to what is currently available in the market.
Can you talk a little bit about what those scales represent? And I understand when we talk about quality of life, early Alzheimer's, the symptoms are almost imperceptible, right? We all forget things as we get older. At what point do we move from being forgetful to symptomatic of Alzheimer's? That's kind of a key question. And I think what you're saying is that the daily life composite score has trouble detecting that exactly where that sweet spot is. How does the second scale not have the same problem?
Yeah. So the second scale is the function in an overt Alzheimer's disease, which means advanced Alzheimer's disease. That's the appropriate measure because it's a very defined on like, can you bind your shoes or yourself, or can you get a cup of tea for yourself, or can you sign a check by yourself? So these are the questions in this score, in this scale. And you can now imagine if you have early Alzheimer, your cognition, which is measured by ADAS-Cog 13 and also CDR, some of the boxes, will be sensitive enough to identify a decline of memory because of, these are different questions about cognitive, exactly features of cognition or identifying objects, or you get a screenshot and pictures and you have to remember them, and you are basically failing those. So these are more sensitive. But the activities of daily living, just I described, like binding your shoes, laces, making a cup of coffee for yourself or tea, or writing a check. These are features you don't need to be very much impaired, and you can still do that very well. And that's what is referred to not being sensitive enough to identify a separation.
So it's very clear to me that, a drug that's effective and potentially disease modifying in terms of rescuing cells and the cell survival mechanism probably has a greater impact on the slope of the disease if you're taking it earlier versus later in the disease. That directly contradicts, from my point of view as, say, the head of clinical, to how do I get a drug approved if I go I have the best chance of showing stability in a more advanced Alzheimer's patient with a p-value than trying to show that early on. Is that true?
That we believe that's correct.
And so, given that that's where you're at now, how does the resubmission of data, and I mentioned, I kind of teased a little bit the totality of the data beyond just patients that were treated in the trial. Tell me a little bit about what reviewers are going to look at to determine that, in fact, you have met the efficacy requirement. And you talked about statistical methodology allowing you to use the secondary endpoints. I believe what you were saying was to use the secondary endpoints as the primary endpoints with a higher statistical validity instead of 0.05. And I would just, I think you mentioned like 0.0125 or something less than 0.05. So put that together for me again because it's so critical here.
Yeah. So going back to now what we are discussing in the review, in the re-examination, is to now move into the re-examination for conditional approval, which means the approval will be given for a drug which requires the following features: that the disease is serious and also life-threatening, which Alzheimer's disease is. Because also I want to point out in the recent data analysis, we have identified a huge unmet need because recent high-profile Alzheimer trial failures like tau and GLP-1 demonstrate Alzheimer's disease is very complex. And blarcamesine's upstream approach of autophagy restoration has the strongest consistent clinical data point from the respective phase 2/3 study. And given this high unmet need, a safe and accessible medicine is now needed more than ever. I'm referring to the high-profile recent failures of Novo Nordisk's GLP-1 trial and also the Johnson Johnson trial with a tau targeted antibodies, and they both failed. And that makes it clear that the aging population and the lack of near-term safe medical solutions requires the consideration for now an accelerated or conditional giving blarcamesine to patients, given the ability that you can now argue that this is a high unmet need in this serious disease. And then on top of it, the requirement is an exploratory genetic subgroup that shows a large clinically meaningful effect. And we also meet that criteria because we noticed that in our pre-specified group of sigma 1 carriers, there was a stronger signal and then for those who had a mutation or for the ITT, the overall population in the trial. And then the third thing is that there's a strong mechanistic rationale linking the genetic variant response to the effect of the drug. And we can confirm that as well because our drug activates the sigma 1 receptor. So if you have a mutation, you don't have a perfect effect. It's a bit like a car with a flat tire does not go as fast as a car with fully inflated tires. And so the fully inflated tire car represents the sigma 1 wild type population, which, by the way, the good news is that it's carried by the vast majority of the patients. We talk about 70%, right about 70. And the last requirement is that there is now a commitment to do another trial in parallel to confirm all the set data points which were described. So you just run a trial in parallel while you allow the patients and the caregivers and the physicians to make the choice to get treatment for because it's such a high unmet need and there's no alternative out there with that benefit compared to the risk. And
A European conditional approval, if you
that
will.
will be the conditional approval in Europe. That's correct, yes.
And what's the timing of all of this?
So the resubmission timing probably is run about 4 to 6 months. So we are basically starting this now. that is roundabout what we have seen in the past from other previous cases. Sometimes it goes a bit longer, but I think that's the estimate I think this resubmission examination takes.
So switching gears to the US, I mean, I've heard a lot of different things from people about US, regulatory submissions and electronic submissions, but it seems from what you're describing that the US process is a little bit more streamlined. I know you recently had some interactions with US regulators. Tell me a little bit about what you think is likely going forward in the US. And I know you can't predict and say that the drug is going to be approved or it's not going to be approved, but how aware are US regulators of the European decision? And does that, in your opinion, influence them? Or is this like a second review for them, even though it's not? But they are not, it strikes me that the US doesn't want to repeat the mistakes, if you will, in my opinion, as an analyst, of what the European regulators have done.
Mhm. And we never had interacted with the FDA other than we had a pre-IND meeting before the trial started. And then recently we were asked to meet them, and we had that meeting in a Type C meeting to showcase them our data in a overview like a meeting of a Type C meeting a schedule. And that meeting is now led to the request from the FDA to ask for the full package to review, to decide how to proceed with an NDA pathway to move it forward, given the unmet need and the highly recently failures and the substantial dilemma that there is also currently, with the existing approved drugs in the US, a limited coverage of patients with allowing them to give them access to a meaningful and clinically meaningful benefit drug, given that the requirements from the monoclonal antibodies, which are right now approved in the US, have limitations of safety. There's a black box warning for them, and also the requirement for that reason to measure looking out for brain bleeding and brain swelling through required regular MRI scans. And the vast majority of the country does not have a MRI center in every little city. So it's hard to basically comply and scale this approach worldwide, nationwide in the US. And this means there's a need still, a huge unmet need still for tapping into that. And we are now proceeding with providing the FDA with this full package in order to discuss what would be the best path forward for addressing this unmet need in the US as well.
So, based on your comments, what I hear you saying is, or what I would conclude as an analyst following the company, is that the US regulators are just not going to have safety issues here. What they're going to look at and say, is this drug having an effect? And does the totality of the data that you can submit suggest that there's an effect? Is that right?
That's certainly a way to look at it. We
Yeah.
believe that that could be a discussion going forward. Yes.
Okay. So if the FDA were to determine, yeah, there's clearly an effect here, could they come back to you and say, we could grant you, we think this is a worthwhile drug, we think there's data here, we would like to grant you a conditional approval and ask you to do another trial? That's one scenario, and I find the FDA to be conservative. So is it likely that they come back to you and say, okay, we like the data. These are the parameters we'd like to see in a second trial?
I don't know, but I think that the request from the FDA was to take a look at the data points, the totality of the data, and make an assessment how to proceed and advise us how we should proceed.
Okay.
And that's where we are. So
How long will it take for you to submit the data package to the FDA for them to review it and come to a decision point?
it will be a question of, I don't know how long, but it will be relatively timely. So I would
Couple of months, so like a quarter, 3 months,
probably,
reasonable.
I think that's reasonable.
So, Chris, one of the things that I think differentiates Anavex from a lot of the microcaps I cover is that the company has cash. What was the last reported cash balance?
So the last reported cash was in the quarter call, and it was 120 million in the bank. And we always keep that number run about around to have 3 or more years of cash, including all the trials we are planning to do. And that is without any debt. So
Good.
it's
And one of the things that really like about you is you're very dilution sensitive, right? You don't want to just, you're not gonna uh, I'm not going to wake up one morning and find that you've done some crazy dilutive deal. You seem to understand what you have. You have the cash runway to see it through to the next series of inflection points. And the goal would be on positive news, raise capital, and either commercialize or partner the drug. Is that about right?
we want to be as mindful and fiscal responsible as possible. That's correct.
Good, good. Um, we wanted to keep this conversation brief this morning. One of the things that I'm thinking as an analyst is, research is going to move towards a more interactive approach. Just so for people who are listening, this was unscripted. There were no pre-questions. This is really, you're hearing directly from the CEO what he thinks, what he anticipates is going to happen. And I think this is the future of, from my point of view as a research analyst, this is the future of research, right? It is building a relationship between the company and investors that want to own the stock. And as you know, Chris, I've been in for a long time. I believe it. I have personal reasons for wanting to see this go through that have to do with health and wellness of people, not financial reasons. And so I am absolutely pulling for Anavex 73 to get to the marketplace. Thanks for sharing with us. I'm sure you have a lot of good meetings planned for San Francisco next week.
We do. Thank you, Jason. Absolutely. And a reminder, we are really doing this for the patients, and we are really giving our best to move forward and help patients with this unmet need.
All right. Thanks, Chris. Nice seeing you. Talk to you
Thank
soon.
you.
Bye bye.
Bye-bye.
Thank you.
Thank you.